Thank you, Rob. Good afternoon, ladies and gentlemen. Thank you for staying for the last session of this two-day seminar. Thank you to the organizers for inviting me to speak here. It is a little bit difficult to start a talk after the last topic was how expensive therapeutics are, when the general perception about imaging is that it's really, really expensive. And I'm not going to argue that imaging is not expensive, but I'm going to try to make an argument that perhaps this is an upfront cost that down the line may save some of the cost that not necessarily result in good patient care. So PET imaging, how can it help identify tumor heterogeneity? Briefly, just a couple of slides about what is PET. And then I'm going to focus on a few areas of research in breast cancer imaging. And lastly, a few slides about why is it not easier to bring new radiopharmaceuticals for imaging for cancer patients. So PET. P stands for positron. So positron is a positively charged particle that is ejected from the nucleus of an atom, travels a little bit until it meets an electron, and the result of that interaction is two rays of light that get detected by an electronic equipment that is a scanner. And the most typical, the workhorse of nuclear medicine imaging for PET is FDG, fluorodeoxyglucose, which was initially developed as an anti-cancer drug with the idea of blocking glucose metabolism in cancer cells. It worked great in cell cultures and small animal models. However, when tested in dogs, it put those dogs into coma because their brain were taking up FDG as well. We don't put our patients into coma, as you're fully aware, because we give nanomolar, picomolar amounts of this radiotracer. But fluorodeoxyglucose stands for glucose, where hydroxy group in second D position was replaced by fluorine 18. And what happens is that the GLUT1 through GLUT5 receptors on the surface of the cells do not separate this from regular glucose, and they take it inside the cells, preferentially based on their glucose metabolism. A hexokinase will phosphorylate this. From there, it will be recognized as foreign, and it will not undergo any of the other metabolic cycles. The glucose 6-phosphatase has a less affinity for this phosphorylated FDG, and therefore effectively traps it inside cells, resulting in dramatic cases like this patient with metastatic melanoma. In terms of the actual scanner, this is the first component of the exam, a simple CT scan where the patient sits on the gantry. And a CT tube rotates around it, generate X-rays, and detectors on the other side of the patient identify what is attenuated, what is not attenuated by various organs inside the body. The next step is for this to go into the PET component of the study, and while in classical radiology with X-rays, we detect what comes from the other side of the body. In nuclear medicine, we detect what comes from within the body. So we inject small amounts of radioactive materials that get incorporated into various processes inside the body, and then by summing millions of these events, we're able to create images. So in very, very brief, these are some basic principles of positron emission tomography, or PET. So now let's talk a little bit about a few targets of research for imaging breast cancer patients. One that is developed and worked on extensively here at Stanford is imaging angiogenesis. And I'm going to show you some examples of that and our progress with that probe. Others are imaging estrogen receptor as well as HER2 receptor, so all of them related to breast cancer. Angiogenesis is a process that has been advocated for a long time of being intimately involved with cancer development, metastasis, and if you can block angiogenesis, in theory, you can block cancer progression. The integrin alpha V beta 3 is the downstream marker of this process, and in theory, anything that will affect anything upward of integrin alpha V beta 3 would produce a change in the levels of integrin alpha V beta 3, and therefore, if you can image those levels, you can perhaps select patients for targeted therapy or evaluate early response to treatment. Because of this theory that has been embraced by many groups, there were a multitude of PET radiopharmaceuticals developed for imaging angiogenesis by surrogate biomarker integrin alpha V beta 3. On the top left, it's a monomer, galacto-RGD. They all include this RGD, arginine, aspartic acid, and glycine, because they have a high affinity for integrin alpha V beta 3. The RGDK5 was the code name of the product developed by Siemens. It worked very well. Unfortunately, the cost of tracer development were so high that Siemens decided to sell their molecular imaging branch, and it's currently on hold. No company had picked up further development. Fluciclotide is the commercial name of the product developed by GE Healthcare. That one also was in a few clinical trials, but unfortunately they also decided that the cost of tracer development is too high, and therefore they stopped development. FPPRGD2 is based on the drug silengitide that was in phase 3 trials in GBM, and this FPPRGD2 was developed here at Stanford through all the phases of cell culture, small animal imaging, toxicology, exploratory IND, conversion to a full IND, and now phase 2 trials in patients with a variety of malignancies. So these are results from a first pilot phase study, patient with breast cancer, either newly diagnosed or newly recurrent disease. And what I want to show you to draw your attention to the fact that the uptake, so the semi-quantitative analysis of what ends up in the tumor in biopsy-proven lymph node metastasis, in bone metastasis, and in other metastases was much higher than the uptake in benign lesions. So this was very encouraging for us because that's one of the first things that we want to see, that you have an imaging agent that is specific for processes that identify cancer in this case and is not taken up by other normal structures or things that are, for example, inflammatory. And perhaps there's no better way to illustrate this than with a few images. The first ever cancer patient studied here at Stanford with this integrin alpha V beta 3 agent was a patient with lobular cancer, which is one where we know that the standard of care FDG does not work very well. And if you look, these are maximum intensity projection image from the RGD tracer and here the FDG. The primary tumor, at least part of it, demonstrates intense uptake, suggesting that there is angiogenesis going on. And to the surprise of the clinicians as well as ourselves, there was a sub-centimeter lymph node that was biopsy proven to be metastatic disease in the right axilla, as well as a sclerotic lesion in the thoracic spine that was also biopsy proven to be metastatic disease. And all of these were unknown to the clinicians. This is an example, and it's a perfect illustration, in my opinion, of the heterogeneity of targets we see in the same patient, because if you look at the middle rows, the primary lesion does not have a high amount of FPPRGD2 uptake, while the same lesion demonstrates high glucose metabolism. While the bone metastases in the pelvis have high uptake of FPPRGD2, suggesting high expression of integrin alpha V beta 3. And if that's a correct theory, then high angiogenesis at that level. So I would argue that if you give this patient a drug that targets angiogenesis alone without targeting other components of tumor heterogeneity, the end results are going to be what has been shown by multiple randomized trials, that adding Avastin to standard chemotherapy does not improve overall survival. Yet we all have anecdotal examples of patients who responded well to Avastin, and perhaps the result of those large trials is because there was no appropriate patient selection prior to inclusion in these trials. Why would someone get a drug if the target of the drug is not present? So, that's food for thought. Also, why should you continue giving the drug if, let's say, at 1 week after starting it, you can determine if that's going to work or not? So, as I said, imaging can be expensive up front, but perhaps given the cost of drugs like bevacizumab, this may save money to the healthcare system overall. A very interesting work was done by David Mankoff and colleagues in Seattle, where they labeled estradiol, so they looked for estrogen receptor expression. And this is a classic example in the imaging literature. This is patient A on top. This is a new agent demonstrating multiple bone sites of disease, metastatic disease, demonstrating high estrogen receptor status. Some of these had FDG uptake. Post-therapy, FDG demonstrates normalization of uptake in most of these sites, which is something that we would interpret as good response to therapy. On the bottom, different patient, no uptake in the skeleton. However, this patient on FDG has uptake, bone metastasis. This patient received the same treatment as one above, targeting estrogen receptor. And I think it's no surprise that this patient did not respond to treatment because there was no target of the drug. Similarly, there are many groups that are currently working on either antibody or antibodies targeting HER2. And this is another example from Europe. You'd see that many of the examples in medical imaging literature come from Europe or from Asia and not from the US. The reason for that is that because our regulatory environment is much, much tighter here, and it costs much, much more to develop a new tracer and take it through all the phases of development. But in this case, 3 different patients. So in patient A, there are liver metastases as well as some bone metastases that have HER2 receptor status positivity. Here, there are several bone metastases, and here, there are multiple bone metastases. So if you can collect information about various targets of drugs, in my opinion, although it will be expensive, you will be able to tailor therapy and give the appropriate drug to the appropriate patient and stop giving medication that don't work to patients who are unlikely to benefit from it. And here, just to illustrate the fact that some of these findings that were seen on PET alone were later confirmed by imaging findings with MRI, such as a brain metastasis and multiple bone metastases in the spine. So what are the challenges for us for clinical translation of new molecular imaging tools? Well, it is an extensive drug development process, and the yields in the long run are not very rewarding for the companies that get involved with this. In fact, the more specific an imaging agent it is, the smaller the market for that company. So really, there is no financial incentive for the big pharma to get involved in drug development unless they understand that they can have an imaging compound that can accompany their therapeutic agent and really make for the drug to be much, much more effective. However, it turns out that convincing executives in a pharma is not the easiest thing because they're seeing very much along a certain model that has been around for decades. And bringing imaging in drug development is something that can be seen as a disruptive technology. The FDA scrutinizes these tracers, although they're given in nanomolar amounts, the same as other drugs. So you have to go through very similar processes for drug testing and development. And what makes it really worse is even when you get FDA approval, CMS decides to not reimburse for some of these. And the classic example is the race to develop a beta-amyloid imaging agent. 3 major companies worked on it. They developed it. All 3 are FDA approved. Reimbursement is minimal, if at all. So that's no incentive for any of these companies to continue to develop drugs because they invest millions, if not tens or hundreds of millions, and the end result is that they get FDA approval but no reimbursement. The challenges also rely with the sites where imaging tests are done. We're fortunate here at Stanford to have resources that other institutions don't have available, but we need, in order to do such advanced clinical research, you need to have all the facilities, including production of these radio tracers, as well as institutional regulatory bodies that are willing to take some risks. Sometimes you can get away without filing an IND if your radioactive drug research committee is willing to believe what you have proven in preclinical studies and allow you to do biodistribution studies in up to 30 patients. That will save a lot of money. However, our experience is that when we converted the FPP-IgD2 from an exploratory IND to a full IND, although we had tox study for the EIND in a small animal species, and there were no toxicity in 15 cancer patients, we were asked to do a second small animal study prior to the full conversion to an IND. So sometimes the rules can be really prohibitive from tracer development and translational research, bringing new things to patient care. I just wanted to illustrate, before I end, we have a full ability here to conduct clinical research, translational research, starting with things in the radiochemistry, Lucas expansion, where many of these products are initially tested, cell cultures and small animal studies, in the Clark Center, where biologists, chemists, physicists, etc., come together to create new and exciting products, and then a new molecular imaging clinic on the second floor of the hospital where we can test anything that should be brought to man in a safe environment with very modern and up-to-date equipment. So I like to quote this from the British Medical Journal, and I think it applies to many things. In brief, it says that although the parachutes were never tested in a double-blinded randomized placebo trial, nobody will contend their utility in saving lives. So, from our perspective, when we see many of the things that have been done for a decade in Europe, and then we cannot use them here because they didn't undergo FDA approval, it's just a matter of extreme frustration. And so I'm sure that's something that's reflected in other specialties. But with that, I would like to thank you, and I hope that I kept you awake for 15, 20 minutes. Thank you very much.
Thank you very much, Andrei. Any questions? Yes, in the back.
Yeah. How many lesions did you find differences in uptake of the radiopharmaceutical? And also, the alpha V beta 3 is expressed both on vascular cells and on solid tumor cells. So, can you answer the question?
So the questions are, A, how many lesions were found with one tracer versus the other? And honestly, I don't remember off the top of my head, but there were differences between the two. There were some overlaps, but there were clearly differences. Things were seen with one and not with the other. The other thing is whether integrin alpha V beta 3 is expressed on normal vasculature. And yes, it is. We see it in wound repair, in tumor vasculature, as well as in tumor cells.
No, I'm referring to the expression of alpha V beta 3 on solid tumor cells.
So there are multiple experiments, including studies done here by neurosurgery with Dr. Harsh, who looked at GBM and other tumor types that have shown an overexpression of integrin alpha V beta 3 on tumor cells.
Beverly?
So, Andrei, we've heard a lot about imaging, not about imaging, but about immune responses and how effective they're going to be in various tumors. Is there some way to follow immune response with imaging modalities? I know it's been done in small animals. How about in man?
So there are many efforts, and Sam's group here is working on imaging inflammatory changes and what component those might play in immune responses. There are also imaging agents that are specific to the target. So for example, the one that I'm involved with in lymphoma, we usually do an FDG scan after one cycle or two cycles to predict what will happen at the end of treatment. At the end of treatment, sometimes you have non-specific uptake. So with that in mind, for CD20 positive lymphomas, we did label rituximab, hoping that that in turn will be a more appropriate way to evaluate response to treatment at the end of therapy, because if there's no more CD20 positive cells, then you don't have a chance of having inflammation as seen with the standard of care FDG. So there are many projects in work. What I would hope to see is that more of them that we see now make it to the clinic.
Excellent presentation. You made a very good point on Avastin and its lack of effect or effect. Usually patients are committed to the Avastin until they progress. That's very expensive and probably not the best approach. How fast do you think on imaging we could see a response on tumors that are positive for vasculature and neovasculature?
So we have more data on this in patients with GBM. Where out of 17 patients enrolled so far, 7 had pre-Avastin, 1 week post-Avastin, and 6 weeks post-Avastin. And majority of the patients actually had a minor decrease at 1 week. The 2 or 3 that have a decrease of more than 50% at 1 week, so far one of them has 27 months of follow-up with no changes on MRI, and the other ones, that's all the follow-ups that we have, one of them is at 8 months follow-up, the other one is at 6 months follow-up. Unfortunately, the other ones who did not have a large amount of decrease at 1 week had the common course of disease with relatively stable disease for a few months, followed by rapid progression and death. I don't know if we're on to something here or if it's some random coincidence. We don't have enough patients, but we do have funds to continue to enroll patients.
I think that would be a very, very important use for a drug that costs several $1,000s a cycle.
Right, and perhaps it opens the door for other research trials instead of having the patients on a standard of care drug that currently, as you said, is given until there's definite progression.